Hello.
Hello. Uh, oh, okay. Uli's on. Okay.
Right. Can you hear me now?
Yes.
Okay, good morning.
Back to the real reason why you're here, which is, of course, Uli's talk, Plant Phenotyping Across Borders. A very short, condensed run through his CV. Uli studied biology in Germany at the University of Bayreuth, did his PhD in 91 on the drought in xylem and phloem transport. He became a group leader shortly before his habilitation, and shortly afterwards he got institute director at the IBG, where he is still being the director until now. But in the meantime, he has gathered a remarkable track record of different chairman position, just a selection of them. You're chairman and vice president of the European Plant Science Organization, EPSO, but also prominently set up in as a chairman in different excellent clusters in Germany and Europe. He is head of or primary chair of the IPPN. He's coordinator of the EPPN, and of course, then also initiator of Emphasis. And Uli, we are very much waiting on what you would have to say today. So I will quickly hand over and stop talking for now. Enjoy the show. Uli, the stage is yours.
Thanks so much, Philip. Can you hear me? Should be okay. If I can get the screen. So can you see the screen now?
Yep.
Good. Okay, so first, obviously also from my side, very warm welcome to this whatever morning, afternoon or evening session, depending where you are. Very happy to give this talk today. The title, as was mentioned by Philip already, is about plant phenotyping across borders. Borders meaning technological borders, structural borders, organizational borders, and I also will follow up at the end of the talk quite a bit on the question of how to set up these kind of networks on phenotyping, what their reason is and what their usefulness is. So I will essentially come back also to the introduction which Philip just gave now a few seconds ago. Before doing that, I would actually like to first show you where we are located from where you're hear this voice now. We are in the western part of Germany, very close to the Dutch and Belgian border. Essentially we are running a center which is a multidisciplinary center for plant science, for sciences in general, and plant science is a very small part of it. However, we are doing quite a bit in different dimensions. We are looking essentially for research to improve resource use efficiency in plants and to optimize the biomass. And we have 6 research fields at the moment, research themes which we are following. One is about root dynamics and shoot dynamics. So we are interested in how roots work and how shoots work and how they are contributing to improved resource use efficiency. A third part is looking for alternative biomass. So there's a lot of algae work here. There's a lot of work in perennials, which we do here. Bioinformatics, cell walls, and secondary metabolites is an issue. And then the 2 parts which are very essential for our work for phenotyping. On the one hand, we have founded the Uli Plant Phenotyping Center quite some time ago now, which has the target to quantify plant phenomes at high throughput and non-invasively. And we also have a group which is focusing on enabling technologies, so technologies which are not off the shelf which we develop and which we are using for essentially plant phenotyping and in these other 4 domains. However, even having this quite large group of people, so we are roughly 170 people here now, we also are linking very much into, oops, it was too quick, into networks. Philip already mentioned the different excellence clusters. We have excellence cluster here on robotics, robotics. There's another excellence cluster which is more on plants and plant and environment interactions. We're also running a more applied research in bioeconomy, structural change in the region here, where we are doing more bioeconomy work, and also in the Bioeconomy Science Center. And there are these different phenotyping networks, which I'll come back later on, because these networks are very important for us to, on the one hand, spin out what we do here, but also to take in information and opportunities from outside in order to deliver what we actually want to achieve. Let me start with the question, why is phenotyping so important, and what is the important element of plants, why is so important element of plant and agricultural sciences, and actually also beyond, increasingly more important for the society. And I hope that the video works now. I'm not sure, can you hear the sound?
No, we cannot.
You can't? Can you or can't you?
No, sorry, Uli, we are not hearing the sound.
Okay, so I'll just essentially, you see the video, you see that plants are important for feed, and a lot of bio-based products and renewable energy. So this is what we call alternative biomass, and we are exposed to many challenges. Climate change, very important one, with many different dimensions, from heat to cold to flooding to drought, which really causes yield losses, and obviously also there's a lot of food insecurity linked to scarcity of natural resources or disease, as we have seen before, water or nutrients. And we have to face that, so we have to study how the interaction is between plants and the environment. And in order to do this properly and with high throughput, we need to have phenotyping to understand and to then develop this together with genomics in order to breed for better varieties, which are more adapted to the future situations, also to future situations in the environment, using deep phenotyping things. And I'll come back to that in a minute, that you can see what we are actually doing in that context. And combining all these different technologies is very important to monitor plants in different dimensions, its growth, its function, its structure. And this is essentially what is also the target for emphasis. And I'll come back to emphasis at the very end as a European research infrastructure, which we are presently building to integrate and to bring together different opportunities across Europe. So essentially that was the content of that talk, so as a kind of introduction. And I would now like to come back to what phenotyping actually is with respect to where it actually plays a role. If you look into the breeding process, from design of crops to genetic resources, to pre-breeding, to the genetic analysis of entire populations, to the breeding process itself, so the selection process and the field-controlled environment, which is going on, up to even the management, or increasingly more also the management with respect to precision agriculture. In all of these different parts of the process for breeding and plant management, we need to have information about how plants perform, how plants grow, how the structure is, what the function of the plants is. And therefore, in all these different domains and different process parts, we need to understand what the plant is doing and how and we want to have quantitative data on that. And therefore, phenotyping is a very broad field. It's very diverse. And in each of these different dimensions, there are different challenges which we have to address in order to deliver to the outcome which is done. So essentially, phenotyping is a tool, which is a tool delivering to many of these different aspects. And therefore, it's very diverse, and it has not a single perspective only. So where are the different opportunities and needs, and obviously also challenges which we need to do there? On the one hand, as you have seen before, and you know that we have a lot of activities there which require image analysis. There's a lot of machine learning, a lot of automation, and robotics is needed for sensing. Big data is coming in more and more because we're generating big data, and data need to be exchanged, and we're generating a huge amount of data. And at the same time, this is not just for the technology, but it's also meant to integrate the omics with other omics, so with the genomics, with proteomics, with transcriptomics, with fluxomics, so many omics which we have in other parts of plants and agricultural sciences. Obviously, it's not only in-house, it's also about different time and space scales. So we have, on the one hand, very small scale phenotyping, looking into cellular processes. On the other hand, we're looking into whole crops and fields in the field. We have different time domains from milliseconds to years. So there's a wide range of different things which we would like to quantify. And we obviously have to integrate the biological part as well as the environmental knowledge about the entire life cycle. So from the seed to the seed and back again to the vegetative phase, to the generative phase, and back again. And so this is also linking a lot to aspects like epigenetics, for example, where we need to know very well what has been happening before, and mostly quantitatively need to understand what's going on there. And this is also actually the starting point which I would like to address first. The first thing, which is, from my perspective, pretty neglected at the moment, is phenotyping of seeds, because that's the starting point of everything. And obviously, seeds are very important on the one hand as the yield, the harvest, which we want to have at the end of many crops. On the other hand, it's also very important for the quality of the research which we do. So we set up some time ago a so-called phenoseeder, which is a robot which is analyzing and quantitatively estimating the number of different traits, which you can see there on the right. Don't go into details there, for individual seeds. These individual seeds can range from Arabidopsis, and you see up here a small, very small dot at the beginning of this tip, up to things like maize, where we can, with this system, can actually work through these different traits. The system can handle, collect the seeds. It can sort the seeds, which is very important because we want to have a subset of the seed batch, which is most characterizing for the seed lot in itself. And you can also put this into a more intensive analysis of the seed perspective and seed physiology itself. We can go, as I said already, from Arabidopsis to maize. We have a wide range of different species which we can work on there. We can also, in the future, we are planning to have the analysis of the internal structure. That's upcoming pretty soon now, and then we can have, for example, differences between endosperm and embryo in order to look at the quality of the seeds and the background from that side. With respect to phenotyping, this is a very important issue because, as you can see in the top part, this is a kind of frequency plot of seed qualities, projected areas, volumes, and mass. And as you can see, this is a very wide range of different qualities here. And you can easily imagine that if you take a seed which has double the size of another seed, then you have quite a distance between the different starting point for the plants before they go into the phenotyping facilities. Therefore, selection of these seeds is a very good opportunity to reduce the statistical noise or the noise which is in the material which you have. On the other hand, you can also quantify the statistical diversity of seed batches, as you can see up here. And this is also very important for not only for the experiment, but also it can characterize post-harvest, for example, seed populations, which in itself can be a very interesting phenotyping trait, which breeders and scientists can be interested. This system is also very important for seed physiology. I already mentioned things like germination or epigenetics. And we can also put it into seed bank background so that we can learn more what the qualities in the seed banks actually is. But we are not only obviously interested in seeds, we are also interested in what's going on in the plant itself. And now I will run you through a number of slides, which is essentially showing what is possible below ground. So phenotyping of roots, a very important part. Here still we have to learn much more than what we know from above ground. I still believe that the principal understanding of what the root does below ground is at a very early stage. I just had a number of meetings the last few weeks with breeders where they essentially said, I would like to have drought-resistant roots, and I can just tell you this is not easy. There are many different droughts and many different adaptations, and it's a very tight interaction between the environment. And roots live in a very special environment, which is this not only special with respect to spatial and temporal patterns, it's a chemical structured environment. There's a lot of mechanical properties there, which makes it quite different from above ground. And often what we see in roots, in the root system and how it behaves in the soil is more reflecting the soil properties than what the root actually has, and because the root is very much responding to the soil properties. And therefore, it's obvious that this very tight interaction between the root and the environment plays a major role, and that we can only learn limited limited things from these kind of artificial systems in pots and petri dishes. But on the other hand, obviously, they also give opportunities. So I don't say the one or the other, but it's very important if you want to extrapolate to real world situation, that we have to understand what roots are doing there and what kind of physiology is happening with roots below ground. And in order to do this, obviously the major obstacle for this is that we cannot see through the soil, and normally with our optical systems, with our eyes. And therefore, we started some time ago with using tomographic systems. And there are a number of tomographic systems out there. There are nuclear magnetic resonance imaging systems, and you can see some of them on the left. There's also, these are systems, MRI systems are things which are essentially measuring the contrast based on the availability and mobility of protons in water, and thereby give good information about the general setup of them and the structure. They can also be used to study fluxes, but it's more complicated. A second system which we are also interested in running here is PET, positron emission tomography system. This is based on short-lived isotopes measuring gamma rays, which are easily penetrating through the soil and through the plant, and therefore can be measured from the outside. And we combine the two of them in order to look at the dynamics and the structural heterogeneity of plants while they are fruits, while they are sitting in the soil. And just a few words on the PET system. So obviously on the left side you see here, oops, that's too fast. You see a cyclotron. The cyclotron is needed in order to produce 11C, which is a short-lived isotope which we can get from the system. It's highly radioactive, and when we can apply that, just trying to get this back, we can apply that to the plant as 11C CO2, and this is then taken through the plant, and we can watch with these kind of systems. And this is a small PhenoPet, a new one which we just built. We can see where this actually goes, where the carbon actually goes inside the structure which we determined before by the MRI. And this is kind of examples for that. On the left side here, you can see how the system looks like. This is maize sitting in soil, and then you can see how the carbon actually flows into that soil, into the root. You can see that not every root receives soil or receives carbon, and it's a quite dynamic process to deliver carbon to below ground. We can also go to longer time scales, day-to-day time scales here, like where you can see how a root system is actually developing over time. And on the right, you can see that this is also possible to give quantitative data on the structure. In this specific respect, it's about diameter of the root system in different parts of the root system. So all of this is possible with MRI. So it's a very flexible system, and in combination with PET, we can get a lot of activity reports of the root below ground. But this is obviously not a high throughput system. Therefore, we have alternative systems, additional systems, which are automated, for trait identification. And we do here a lot of work in the past already using these kind of systems here, which is a rhizotron system, an inclined perspex system, where you can look at the root system, which is growing at the bottom of this transparent screen, which can be monitored from the plant by cameras in that system. We have a number of different systems for doing that, of smaller 72 rhizotrons, a bigger one 250 rhizotrons, and we are now coming up with a new one which has 1,000 to 2,000 rhizotrons available, because this allows really to go to high throughput. These are the kind of sequences which you get with that from the same plant, so it's repeatedly the same information from the same plant. And the big system, you can see a picture here over here, is a kind of plan how it looks like, but as you can see really on the left side that it's already installed. We have a number of imaging stations up here in this floor plan, and we can expand to more of them, and there are quite a number of different systems in there. We then delivering the rhizotrons, these blocks here with HEVs, so autonomous driving vehicles, going to these platforms, unloading them and reloading them and bringing them back to the system. So it's a quite complicated large-scale system which we are just finally commissioning. We get a lot of traits, shoot traits as well as root traits from the system. So it's not only looking at rhizotrons at roots below ground, but it's also at the same time looking above ground, because obviously there's a very important to have both sides of the plant imaged at the same time and getting quantitative information. But it's not stopping there, obviously, specifically also with roots. There's the real reality is outside. So we have a number of systems which we are running in the field. One of them is a shuffleomic system, essentially a kind of digging out the roots. And then obviously also here we have issues with measuring in high quantities. So we have built systems where we can rotate these root systems, in this case cassava, do kind of extraction of the information from the root system, and then put this into a bigger picture. And the bigger picture is coming to this part then, that we have we feed all these kind of data obviously into models which give us a much better opportunity to link the different systems, to have a better overall understanding what the system is about. Looking also at kind of virtual experiments about planting densities, for example, or about competition between the different roots. And this gives us a lot of opportunities, which then also feed back into the phenotyping systems which we built, but also in the experiments which we have there. So this is then all together to develop idiotypes, which we think is important for the breeders, to integrate the different phenotyping experiments and to link between lab and field, which is still one of the major challenges which we are facing in phenotyping community as a whole. So this was a lot about below ground. Above ground, obviously we also do a lot of above ground work. This is much more focusing towards the breeding and farming community. And essentially what we do there is we obviously start with a number of traits. We have a number of traits which you want to look at, again, roots, and you see that we use shuffleomics, for example, for that. But we also have structure, we have water relations, we have photosynthesis, which we would like to measure, and there's a number of different sensors, a large number of different sensors. This is just a very small set of sensors which is available, which can give information in order to extract these traits in quantitative terms. At the same time, we need to bring the sensors to the plant, because obviously, in contrast to what we do in greenhouses, where plants go to the sensors, we mostly in the field have the sensors going to the plants, and therefore we have different position systems. I'll come back to this in a minute. We also then run a number of specific experiments, phase systems or common experiments in common gardens, and obviously a very important part of field phenotyping is environmental sensors, from atmosphere to soil to also plant systems which we actually can measure there. There's a number of different systems which we use here. This is a set of different positioning systems. So this is quadcopters, zeppelins, fixed wing gliders, autonomous driving systems, tractors. We also have push bikes, field bikes, and also use them in the breed phase, for example, which I come back in a minute. And we have a number of different sensors from monitoring systems, and this is just the ones for photosynthesis, for lift, laser-induced fluorescence systems, which is a laser system which measures photosynthesis. We have SIF, which is a passive system. We have the high plant and high screen systems, which give us opportunities to measure passively photosynthesis with fluorescence from sunlight. We do this in experiments, in common experiments, in large-scale plots, in our own ones, or in our own experiments. This is the one which we do in Campus Klein-Altendorf in Bonn, together with the university there, with different varieties, as you see here. But we also have a lot of experiments directly with breeders in breeders' fields, and where we support them with our the equipment, or obviously also together with other academic partners. Special experiments, special setups, and this is we also run specific fields, simulation sites. One very dedicated one is called breed phase. This is a phase system, as you can see up here, a free air carbon enrichment site. These rings, from these rings, we are releasing CO2, and so that the CO2 concentration inside these rings is increased to simulate future CO2 concentrations. These phenotyping, these breed phase systems are very special because they also require special phenotyping devices. We want to obviously also have the same full set of different traits which we want to measure there. And one of the recent systems which we have commissioned there is this field snake, which you can see down here. This is a wide-range, wide-reaching platform which is able to drive itself over these rings. That's the reason why it's actually that broad. It has a width of 18 meters, and this yellow platform here holds a lot of sensors, which is then automatically drawn over the different crops and measures all the different things which I mentioned before. So this is a very special new device which we have, and we are just now commissioning the second and third dimension, actually already from this one, because it proved to be very useful in these field phenotyping dimensions. A new direction which we are also following now since roughly 2 years is to include robotics into the whole context of phenotyping. This is on the one hand still based on the monitoring systems using drones, but also these bigger robots. We are also now going to smaller robots. Robots also often in our case obviously is monitoring, but robots also provide the opportunity of interventions of weeding, for example, can be done, or spraying. And we have a lot of data machine learning issues which are linked to these robots and the steering and the controlling of the robots, which will deliver a very new, completely different way how we handle phenotyping in the future, also with respect to breeding, but also offering a very nice new link to precision agriculture. We link that to our understanding of plants, and I showed you before how we do that. And we also have to bring this into a context of the environment as a whole on a broader scale, closer to precision agriculture, larger scale approaches for farmers, and also into the context of environment and how the impact of these machineries is on the environment, and if we can achieve a more environmentally friendly agriculture using that. And the third part, which is only mimicked here on the one hand, on the very right side, is also about the economic consequences, because robots are nice machines, but in the end, we also want to see that they are really delivering on the economic side. And therefore, we also have groups in that excellence cluster which are focusing on the economic value and how to overcome innovation hurdles using this for these robotic applications. And this brings me to the last part already with the phenotyping networks. So as you saw before, and we are working a lot of networks, we really need to bring different people together. There are technologists, there are users, there are phenotyping experts. So it's quite a large range of different groups which we have to integrate and bring together. And this is an exercise which started already some years ago with essentially the Australian plant phenotyping facility being the first one, then the German plant phenotyping facility network came in, Phenome from France came in. So now we have a relatively large number of national platforms, and there's an increasing number of large national platforms coming up, which are organizing and providing phenotyping opportunities within countries. At the same time, obviously, there is an interaction on the regional and continental scale. There are things like emphasis EPN, which I'll come back later on. There are also continental activities in different parts of the Americas or in Asia, where regional networks work together for different purposes. On the one hand, on specific topics. On the other hand, on providing infrastructure like we do, for example, in EPN and emphasis. And the third dimension is international networks. There are also crop-specific aspects. So, for example, the wheat initiative, we are also working there's also a wheat phenotyping expert group. And everybody who is working in wheat is very welcome to also participate in there. And then the general development of international phenotyping. Sorry, Philip, for the old logo there.
I was just about to mention it.
It's the International Plant Phenotyping Network, which is obviously one of the hosts of this webinar here. So what is the reason for all these different phenotyping networks? So in EPPN, we have the situation that we want to provide access to installations. And there was a first EPPN project, which started 2012 to 2015. It had 21 installations across Europe, and you see some of the links there on the maps below. It was a small startup project with only 14 partners, but proved to be very successful because the target of these EPPN networks is to provide access for groups, for scientists, for industry, which has not access to this kind of specialized infrastructures. So it's not a good use of a lot of investment going into these infrastructures if you only have it for yourself. And therefore, this was a kind of initial spark of the idea of EPPN. That we would actually like to give access to these infrastructures for other people with other scientific approaches. This went extremely well because if you look at this from a perspective of a science manager, you invest 2 million and you get 50 more. It's actually now much more than 50 publications. So it's the range of 80 publications now. So 2 million for 80 publications is something where you go for if you are a science manager. And therefore we went the next step into EPPN 2020, which is still running, as Philip already mentioned before. It has more partners. It has 31 installations across Europe. And if you look at the website of EPPN 2020, which you can also, there will be a web link for that later on. You can find you can browse through all these different platforms. You can, for example, select all the platforms which are providing root access to root phenotyping, all the platforms which provide access to photosynthesis, and you can easily spot where these kind of platforms are. And based on that, the process is that you can then apply for using these platforms, and the European Commission funding that project is actually giving them the funds on the one hand to the users, on the other hand also supporting the infrastructure itself, the maintenance costs, the running costs of the infrastructure, which helps a lot for those people who invest in these kind of systems. And based from this kind of initial experience that this kind of linkage of infrastructures and opening of infrastructures would be very useful, we started a process of emphasis as a European infrastructure for plant phenotyping. And it's plan to form a pan-European plant phenotyping research infrastructure. This is within a process or a framework which is called ESFRI, European Strategy Forum for Research Infrastructure, which many European countries, all EU countries, plus some additional countries use to coordinate their investments into infrastructures and to research infrastructures, and to open these infrastructures for other countries in themselves. So essentially the meaning of emphasis, the mission of emphasis, is that we would like to present and facilitate multi-scale plant phenotyping to analyze the different genotypes. So we are not genotyping ourselves, we are not providing the genotypes ourselves, but opening the phenotyping for other people who have different genotypes, and to provide this in diverse environments and also in controlled environments, and to quantify a wide diversity of traits. And I just mentioned some of them in the talk before. And how is this done? There is a wide range of different phenotyping facilities and installations across Europe. And we are offering the opportunity to develop this infrastructure and to provide access to this infrastructure, to facilities, to services, and to certain resources, which are hidden usually within a national or even a research organization internal situation. And therefore we open this, and we are generating a lot of synergies on the infrastructure level, but also on the scientific level, so that people can much more easily collaborate around these large-scale infrastructures. The plan is to develop these pan-European infrastructures and instrumented facilities in a very detailed way. We are working on this now since close to 3 years, and we are coming to a situation where we're close to launching these activities. We also link the data acquisition from all these different systems at a European level for data information systems to have the opportunity to run different experiments with the same genotypes, different experiments with different environments, for example, at different places in controlled environments in the field, and combine them in order to deliver all of them at a certain quality of information. So quality assurance is a very important part also in that part of data acquisition and data information. And last but not least, we also use this for developing, disseminate, and share knowledge on novel technologies and on phenotyping, on best practice for phenotyping among the phenotyping community, but also spread it into a wider range of groups, into breeders, into users from science and non-academic background. So the benefit of this installation will be in the future for researchers, and for industry. It will also be an opportunity for public sector investors because we obviously would like to have a complementary infrastructures across Europe, so that different countries, different organizations can actually share infrastructure, and you can much more easily convince investors to go in this direction if you have access and provide access to other infrastructures. The society in general, I think the great advantage of phenotyping in many cases for science communication is that we are working a lot with images and pictures, and we can easily illustrate the importance of plant sciences and plants. And obviously science organizations themselves will also benefit from it by using synergies in operation of these expensive infrastructures in the future. There are 5 different areas which we are working on in order to combine them in this phenotyping infrastructure. One is about controlled conditions. This is all the high throughput systems which I mentioned before for roots, for shoots, but also the deep phenotyping activities, tomographic systems, MRI, PET, CTs, which are out there, which we can combine and which we also provide access already today. There are also so-called intensive fields. This is fields like, for example, our breed phase, where we can have very intensive instrumentation, where we have simulation systems, very highly equipped systems, which are giving a well-monitored field conditions for plants and the environment. Lean fields is rather going towards a application driven for breeders and for farmers, where we can have less sophisticated equipment, but more practice related equipment. And with respect to data, we have two parts. One is about the modeling, because we obviously also want to integrate these models into the phenotyping process. And we can deliver from the phenotyping very interesting data to the modelers in order to provide the data which they actually need in order to give meaning to their models and to run their models. And the last part, and I also mentioned this shortly before, is about integrated data and computational services, which are linking the different installations, which are also important to quality control the activities in the different installations, because you want to have only high quality data in your system. So we are running this in a way that we have certain quality levels which different installations can reach and have to reach in order to be able to bring their data into the system and to interlink then the different data which are in there. These are the contact data, and I guess that afterwards you will get these slides anyway, so you can read through these contact data, and if you want to, you can also contact us directly through that. And last but not least, the IPPN. This is now going global. The IPPN with the new logo aims to integrate the regional and globally fragmented activities. We want to enable knowledge, information exchange, and experience exchange, and also hopefully at some stage when we are allowed to travel again, also exchange of personnel between different places, addressing technologies, and also advise decision makers at different levels to not make the same kind of mistakes which everybody of us that did before. So to have a kind of more educated approach when before you really buy something and before you design a new system. It's very helpful to know what is out there already. It's very helpful to know from the community, from people who used equipment before, how it performs. There's in such a diverse field like phenotyping, there's not a single golden bullet which will deliver all the stuff, and therefore it's good to have knowledge and information before you actually build things. And this is a relatively rapidly growing organization. We started with, I think it was 17 or 18 groups at the beginning in 2015. Today we have 44 research organizations and 7 industry members. Essentially, since 2018, we also opened for industry members. Before it was a pure academic organization focused institution. But this group is growing, and it's also open for new members. So if your research organization is not yet here with all these logos, it's very valued to join and also contact Philip on that. And also if you're an industry member, person, you can also think about joining the IPPN. It's for academia and it's for industry, for growers, for breeders, but also for technology providers. So we have a wide range of different groups which are in there, where we are open to collaborate and where it's useful to bring in your expertise together on the table with other experts in the fields of all these different disciplines which are in here. An essential element of the work in the IPPN is the so-called working groups, and this is the number of working groups which we have today. This is about advanced sensor applications. We have also a group about affordable plant phenotyping, so this is things like small smartphone-based phenotyping or phenotyping on cheap devices, which can be very valuable. Field phenotyping is addressing the special cases for field issues, including all the performance from the crops through the sensors to the environment. Forest phenotyping is also a new working group coming up now, because obviously people working in forest breeding, but also in forest management, have also questions about how to quantify the information from their plants, from their trees. Imaging for plant phenotyping is focusing on imaging, because that's obviously a quantitative imaging, which is a very important part of the whole process. And they also have actually started some years ago already about with webinars. So this is also very useful for people who are interested in that domain. controlled environment plant phenotyping is also coming up now with specific approaches for highly controlled systems which are trying to get closer to the field. Food phenotyping and seed phenotyping are already described the importance of that before, and we're also running strongly within these working groups. Another important instrument of IPPN is the International Plant Phenotyping Symposia, the IPPS. And these has been a series of different conferences initially loosely connected, now in a sequence, and we are now already in the range of the 7th IPPS. We had the 6th IPPS last year in Nanjing, and there will be this is a meeting where the international community on plant phenotyping comes together. There are sessions on the research and how to use plant phenotyping and what kind of technologies are there. It's also giving opportunities for technology exchange and industry and academia interaction. And the next, as I said, the 7th IPPS will be in 2021, and that will happen in Wageningen. And they just start their national system there, and it will be very interesting to see their new systems in 2021. So let's hope that we can come together face to face and physically in 2021. Otherwise, I'm pretty sure that we also will have an online version of that, hopefully, or a mixed version. So thanks very much. I hope I could give you a little bit of overview of what kind of technological issues are there and how we try to overcome them, and also how to link different activities, what the reason for all these different networks are. So thanks very much, and I'm very much open to questions if you have some.
Yes, thank you very much, Uli. Can everybody hear me? Yes, I guess so. Uli, can you hear me?
Yes.
Then everybody else can hear you. Can hear me. So we have accumulated quite some interesting questions from the chat, and we would have it like this: if the person who asked the question is still present in the auditorium, then I will quickly read out the question and open the mic for this person, so they can interact directly with you, Uli. So first question came in from Dina Serak. She asked, "Thank you for this interesting opportunity, and I would like to ask, what about African phenotyping crops? I assume that it would be a good idea to have the same opportunity to study for this one like the Chinese one. What is your opinion about this kind of action, Professor?"
So there are a number of different components to that. The first one is we are actually trying since several years to get members from Africa into IPPN, because exactly what you say is Africa obviously has many opportunities and many challenges with respect to phenotyping, with different crops, with different environments, and also with breeders and applications and food security, and beyond that, which are available there. So far, we did not really succeed in that, but we would be very open to reach that. With respect to the use of infrastructures which we have here, we also have some experience with African projects. So we have projects from the Institute, where on the one hand we had in the past, we have projects in a number of different African countries, where we use our phenotyping gear, which is mobile, for field phenotyping in Africa. So we have, for example, projects on cassava, we have projects on teff. So we have, and there's quite a range of different projects which are running there. But we don't have a large-scale phenotyping partner there. Another opportunity which we also had previously in projects which were also funded from the German side in the context with Africa, and we most likely will have one which opens there again soon, is that we offer stipends for people from Africa coming here, where they can then use the facilities which we have here. So for example, we had colleagues here from Africa who were phenotyping banana, bringing their genotypes and doing the phenotyping here. So it's always it's an opportunity, but at the moment we haven't been successful in getting a real partner in Africa into IPPN.
Yes, thank you. Although I have to add that this is on our radar, it's on our roadmap, that especially in developing countries, of course, the benefits of plant phenotyping are valued the most, or the impact is potentially the highest. Yeah, so whenever you feel like it, you can go on our website, IPPN, plantphenotyping.org, and then you will find contact addresses. So just drop us a mail and we'll dive into the possibilities going from there on. The next question is in the same direction. Yeah, it says plant phenotyping at both universities and research stations in developing countries is predominantly characterized by weak or no structural facilities. For example, so that is Damodar Pudyal, if I pronounce it right, sorry for that, is working on habanero pepper for heat and drought stress screening during the vegetative and reproductive stage. But all he's missing is a climate chamber. In situations like these, he continues, how can we contribute to plant phenotyping from the developing world? And this I find an interesting aspect. How can they participate?
So it's a little bit along the line which I already said. I think there is, on the one hand, there are opportunities to link up with countries who have facilities to do that, depending if it's controlled environment. Obviously, if you say growth chamber is missing, that's the thing where it's obviously hard to bring growth chambers from here to Africa or to any other developing country. But there are opportunities and many of the partners in, for example, IPPN, or also on a European scale, we have partners in developing 8 institutions who would potentially be able to provide such kind of facilities, depending on the specific opportunities of the countries and the links between the different countries. So it's not excluded, and I think it would be an option if we get the kind of information what is needed, that we can work on that. Definitely not guaranteeing that it works, but I think that's one option. The other option is to have joint projects. We have in many partner institutions of IPPN and many research organizations the opportunity to link activities through projects. A third part, which I didn't mention before, is specifically this aspect of the affordable phenotyping. Today, it is really possible to get a lot of quantitative data from very affordable instruments. So based on, for example, smartphones, or based on very cheap sensors, which are plug-on sensors, or based on Arduino computers, for example. There's a lot of options which one can use with very cheap devices, which already deliver very good results. So this would also obviously also be, this is one of the targets why we have started the affordable phenotyping group. And therefore, I think there are specific options, and if you have specific interest there, probably also that's what Philip would also say. Try to contact us, and we try to find potential links where we know that some people have options there. Not guaranteeing that it works, but if there are options, we are trying to find them.
Thanks, Uli. Then we have another question by Songyang Lee. It is asked, thanks for the presentation. I saw that crop phenotyping can be integrated into precision agricultural research. Is there any possibility to combine field-based phenotyping and large-scale remote sensing-based climate changing studies?
So that's, yes, of course there are. And I actually took that out of the presentation, because usually in the presentations I would have mentioned that we are also doing satellite imaging and remote sensing from space. But I did not put it in for today because I thought this would go beyond the scope. But obviously, yes, we have a lot of remote sensing, a lot of airborne sensing of crop performance in precision agriculture. And because in many cases there are similar questions, we are also combining them with remote and proximate sensing in the context of phenotyping and climate change. And obviously another aspect which I also did not mention too much is there's a lot of options there also for phenotyping biodiversity. So if you go into a more natural biodiversity background in the field or in-house, you can also learn a lot using these technologies. So it's essentially technologies which you can apply to many different backgrounds.
Absolutely. So plant breeding is only one of the contexts in which plant phenotyping can yield benefits, and of course, yeah, this is just the beginning. We have another question coming in from Brazil by Paulo Herman. Could you tell us a little bit more about the experiences with interactions with industry and breeders? Where's the key point now?
Paolo, you know that's a very, very complex one.
Yeah.
Because there's not one breeder, is not one industry. It's very diverse, and it's always very much, it's also changing over time, because obviously in some years ago, breeders themselves didn't have facilities which were at a very high technological standard. At that time, we got a lot of requests about specific technology or even developing technology. Today, many breeders have technology which is state of the art. Many breeders have options to also invest in that area. So today the questions rather go into how to use systems properly, and how to use systems which are not easily deployed at the moment. So a lot of requests goes to root phenotyping, obviously, because that's still one of the bottlenecks. Data handling, data quality, data assurance is something which is obviously very important. So there's a wide range of different requests. At the same time, with practical breeders, it's also an issue that obviously a lot of the high-tech part, which is used in science background, is just not valuable enough for a breeder. Therefore, a lot of the questions which we have there is to reduce complexity to systems which are available and which can be more easily deployed and deployed at different stages in different places. So it's a very diverse community. It's also the same with industry. If you talk about industry, you can talk about industry in food production. They obviously have different questions compared to ag industry asking about chemicals. So it's diverse. Obviously, it's also very much dependent on different countries and the culture in different countries, how to work between industry and academia. But in many cases today, also a lot of developments in phenotyping already happens in industry, because they have now built up experience in this field, including the non-invasive phenotyping technologies. themselves, which are very valid and useful for them. Last part to that, obviously, it's very much dependent if you talk to small industry or big industry in small breeders or big breeders, because obviously bigger breeders tend to build up their own systems and tend to build up their own facilities and competence, while small breeders usually have to put themselves more into a network with respect to all the use from developing to access. While with bigger companies, they often do development themselves, they often do deployment themselves, they often have, and they have to build their systems into their entire research landscape inside the company themselves. So it's pretty complicated. And many diverse directions which we see there.
Yes, thank you.
Thank you very much, Uli.
The next question comes from 1997, and he wants to elaborate, or he wants to ask a question on root research and the scale of it. I think the mic is open. 1997. Can you precise that in some sort? What do you mean by that? You can speak now. 1997. Okay. Obviously, he's not on the mic, but I have another question in that regard from Jorge Navarez. He asks, I work with stress tests for deficit and excess water. What is the maximum size to do root phenotyping?
You mean by numbers or by sheer size of the plant?
No, I think he means by numbers. But maybe you can answer both questions.
That's again very much dependent on the experiment. So we have systems which have a throughput. If you go to small systems, and you can also work there with kind of not too complex environmental systems. We have systems where you can have a throughput of thousands of plants per day. On the other hand, we have there are systems, if you go for MRI, for example, this is at the moment a maximum throughput of something like 20 plants a day. We're trying to increase that, but the throughput itself, we are a little bit critical about just a number of throughput. Essentially, what you should ask is, what is the capacity? So what is the capacity of crops, of plants, which you can do over a year or so? What kind of experiments? Because if you just ask for throughput, there can be a maximum throughput, which is not telling anything about a real availability. So obviously, this is also very dependent on the different crops and different environmental conditions. What we often do is, because we have a diversity of different phenotyping facilities, that we start with a higher throughput system, go to a more precise system where we have identified the more concrete issues which we see with the roots, and then end up with fully three-dimensional, four-dimensional activities with using MRI and PET. So we rather build this kind of phenotyping chains where we combine different technologies with different throughput and capacity, and with different precision and detailed experimental design which is behind that. Not sure if this answers the question.
Thank you. Next question coming in from China, from Shengpeng Sun. Could you please give a brief introduction about the challenges of 3D plant phenotyping, specifically under field conditions? Any experiences?
Would you like to give it more precise on that? Because I could say something, but I'm not sure, 100% sure if I go in the right direction. So 3D plant phenotyping in the field at the moment is obviously shoot. There is no root 3D phenotyping except of destructive ones. In the field, obviously, anything which where you have sensors is dependent on, if you go for light sensors at least, is dependent of light conditions. Then it depends on what dimension you're looking at, what size of field, what size of plot you're looking at. If you have homogeneous light conditions, if you have a system which looks at a whole region at the same time, or if you have a system which is moving from one spot to another spot, which makes it more difficult to have homogeneous conditions at different places at the same time. So 3D phenotyping in the field is dependent on many cases on the conditions of the acquisition of the data. There is quite a number of different technologies there, which provide options for 3D in the field. On the one hand, systems which go deeper into the canopy, LiDAR things, or other things which are looking at the 3D surface of a canopy or of plant. So again, it's a wide range of different technologies which are there. In many cases, we need 3D phenotyping, 3D measurements in order to interpret properly the functional analysis with respect to reflection or fluorescence signatures, because they very much depend on the 3D structure of the surface. So there's a wide range of, I'm not sure if I answered your question. It's going in the direction which you wanted to ask for.
Yes.
Thank you, Doctor.
Thank you. There he is. Okay.
Thank you, Doctor. And could you please give an introduction about? I found that now it's really hard to get very dense and good quality point cloud and field conditions. I mean, the sensors. So for indoor conditions, I find there's no problem. But field condition is really hard. So could you please provide some suggestions about how to collect dense point cloud under field conditions?
The first thing, if you look at the point cloud, obviously, and that's what I meant with depending on the system which you're looking at. If you have wind, if you have changing environmental conditions, if you have movement of the plant, then obviously this leads to a blurring of the system of your picture, or of your data, if you have a sequential acquisition method. So if you have a method which is giving data at one spot, then obviously the blurring is effect of movement of plants, for example, is much less. And then again, as I said before, it depends on if you want to have the surface of a canopy, because then you could go for stereo or multi-camera systems, or for fast laser systems. Or if you have really the interest in going deeper into the whole process, deeper into the whole canopy, then obviously it's getting much more complicated with overlaps and things like that. It very much depends on the specific question. And if you have a technology which allows you to take images or the data for the point cloud at one spot and at one point in time, that obviously helps a lot to avoid this kind of fuzziness of movements of plants, and also the conditions of all different parts are helping. If you have conditions for that, it also helps. Homogeneous conditions also helps to interpret data.
Okay, thank you very much.
Dankeschön. So we are coming to the last question, and now I'm waiting for the chat for Sven to select one of the last 3 remaining questions to be asked. Sven, do you please have a question for me?
Yeah, just give me a second.
So otherwise I can. I can open the mic because Sudhir Kumar had a question.
Yes.
Sudhir, can you speak?
Yeah. Yeah. I have a question that you have shown so many good work. So how do you deal with the large number of images and the large number of data you acquire? Because we find it's the bottleneck for us. So how do you deal with this? Can you elaborate a bit about this, please?
Okay, if I understood the question correctly, it's the question how to deal with the large number of data which we acquire if we have images. So obviously there are different ways to reduce the data to information. In the end, we are interested in information, not just in piling up images. So depending on what you're doing there are on the one hand things where you could do directly at the acquisition. So if you know exactly what you want to do, you can have systems which are already compressing data to or extracting data from during the imaging process to the real data which you really want. The second point is that obviously we want to have large scale data on the one hand for precision, but in many cases, to be honest, I think at the moment people don't think too much, think enough about the amount of imaging pixels which they actually need, and just take the most biggest picture which you can get from the most biggest camera which you can get. In many cases, my guess, if I look at data, would say that a significantly reduced number of pixels or a less high resolution camera would also have done the job. So today, in many cases, people just take the size of the camera, the maximum size of the camera or resolution of the camera, which helps in many cases, but in many cases it's not actually needed. So that's also a reduction. So that's probably also a more general point. I usually tell people, please think first before you use machinery, and not just use the most fancy machinery which you can find because you have a question for which the machinery should help you, and not just the machinery which is the most important. So obviously another point is then the data acquisition. We have most of our data which we handle today are either handled on cloud computing or on the machine itself. So there is a lot of options there from the compute power, which is good enough to deliver the result. We also have batch processing in systems. In very few cases, but it's more kind of for technology development. We also use supercomputers for doing that. But this is more for identifying the most, the fastest algorithms. So we are not running the systems on the supercomputers, but rather trying to do the algorithm development on there to test more different opportunities. Obviously, in that specific case, if it's about images, it's very recommendable to talk to people who are very used to image analysis, and I can only recommend there to also go back to the webinars which have been done by the working group on imaging for plant phenotyping. They have, I'm not sure if they had one on compression, but they definitely have people who know a lot about this specific aspect. Okay. So very welcome to also contact them.
Thank you so much.
Philip, you're there?
Yeah.
Philip? Did we lose Philip?
Yeah, Spanish speaking seems like.
Sorry about that. I was speaking for a minute or so. So thank you. Thanks to, first of all, to our speaker today, Professor Uli Schurr, for giving us this very, very comprehensive and state-of-the-art overview on the plant phenotyping and all the plant phenotyping networks as well. Thanks for the advertisement, Uli, and also thanks to the audience for being there and giving us some interesting questions. If you look on the screen right now, you see what will be upcoming in our next webinar on June 12th. It will be by Professor Menachem Moshelion from the Hebrew University of Jerusalem on functional phenotyping of plant responses to abiotic stress. Hope to see many of you in 2 weeks again, and I'm already looking forward to this.
Thanks very much also from my side for all the questions, and thanks for organizing that. Thanks. Bye.
Thank you. Bye-bye.